What slime mold serves as a model organism in developmental biology?
Dictyostelium discoideum has been studied as a model for the developmental biology of complex organisms. Under optimum conditions, this organism lives as individual, amoeboid cells. When food is scarce, the cells stream together into a moving mass, resembling a slug. That differentiates into a stalk with a spore-bearing body at its top. This structure releases spores that can grow into a new amoeboid cell. The development from identical, free-living cells to a multicellular organism simulates many of the properties of more complex and complicated organisms.
Who was awarded 2 Nobel Prizes for DNA sequencing?
Frederick Sanger, 1918, won the Nobel Prize in Chemistry in 1958 for his work on the structure of proteins, especially insulin, and shared the Nobel Prize in Chemistry in 1980, along with Walter Gilbert. 1932, for contributions in determining the nitrogen base sequences of DNA. This method was later referred to as the Sanger sequencing method for reading DNA.
What is a population?
A population is a group composed of all members of the same species that live in a specific geographical area at a particular time. An example of a population might include all the gray squirrels that live in a certain urban park. The areas occupied by a population could include the small area, measured in square millimeters, occupied by bacteria in a rotting apple to the vast areas of ocean. Square kilometers, that include the territory of migrating sperm whales. Population ecology is the branch of ecology that studies the structure and changes within a population. Studies of specific populations will indicate the dynamics of the population. In terms of active, ongoing growth, declining growth, or stability.
Is all DNA found in the nucleus?
In addition to the nuclear DNA of eukaryotic cells, mitochondria, an organelle found in both plant and animal cells, and chloroplasts, found in plant and algal cells, both contain DNA. Mitochondrial DNA contains genes essential to cellular metabolism. Chloroplast DNA contains genetic information essential to photosynthesis.
How were fungi involved in World War I?
During World War I, the Germans needed glycerol to make nitroglycerin, which is used in the production of explosives such as dynamite. Before the war, the Germans had imported their glycerol. but the British naval blockade during the war prevented such imports. The German scientist Karl Neuberg, 1877 to 1956, knew that trace levels of glycerol are produced when Saccharomyces cerevisiae is used during the alcoholic fermentation of sugar. He sought and developed a modified fermentation process in which the yeast would produce significant quantities of glycerol and less ethanol. The production of glycerol was improved by adding 3.5% sodium sulfite at a pH of 7.0 to the fermentation process, which blocked one chemical reaction in the metabolic pathway. Neuberg's procedure was implemented with the conversion of German beer breweries to glycerol plants. The plants produced 1,000 tons of glycerol per month. After the war ended, the production of glycerol was not in demand, so it was suspended.
How many kinds of mushrooms are edible?
Among the basidiomycetes, there are approximately 200 varieties of edible mushrooms and about 70 species of poisonous ones. Some edible mushrooms are cultivated commercially, more than 844 million pounds. 382,832 metric tons, are produced in the United States each year.
What is a mutation?
A mutation is an alteration in the DNA sequence of a gene. Mutations are a source of variation to a population, but they can have detrimental effects in that they may cause diseases and disorders. One example of a disease caused by a mutation is sickle cell disease, in which there is a change in the amino acid sequence. Valine is substituted for glutamic acid. Of two of the four polypeptide chains that make up the oxygen-carrying protein known as hemoglobin.
What is a nucleotide analog?
Nucleotide analogues are compounds that look like the nucleotides in DNA. They are used as antiviral compounds because the nucleic acids assembled with these analogues fall apart. Therefore, the viral genome cannot be copied and the infection cycle is broken. AZT is an example of a drug that interferes with HIV's ability to replicate its genome. By substituting azidothymidine for thymidine, thus terminating viral DNA reproduction.
What is the function of each of the different tissue systems?
The vascular tissue system consists of two kinds of conducting tissue, the xylem and the phloem. The xylem conducts water and dissolved minerals. The phloem conducts carbohydrates, mainly sucrose, hormones, amino acids, and other substances for the plant's growth and nutrition. The ground tissue system consists of three cell types: parenchyma, collenchyma, and sclerenchyma that have relatively thin walls and living protoplasts for storage, photosynthesis, and secretion. The dermal tissue system is the outer protective covering of the plant, including the cuticle.
Why do you need protein in your diet?
Of the 20 amino acids used by humans as building blocks for proteins, 8 are essential. In other words, our bodies cannot synthesize them, nor can we survive without them. Luckily, all of the essential amino acids can be acquired by eating animal meat. Complete proteins, and/or certain plant sources, complementary proteins. By combining grains, such as rice, and legumes, such as beans and peas, one is able to consume adequate daily amounts of protein. The recommended daily allowance, RDA, of protein for healthy adults is 0.4 g/lb, 0.8 g/kg, of body weight.
What is the difference between inducible and repressible operons?
Inducible operons are used to produce proteins only under specific conditions. Most bacteria use glucose as their main energy source. Therefore, the unusual arrival of the milk sugar lactose in the cell turns the operator switch to the on position, which allows transcription to occur. Repressible operons have operators that are always on, transcription occurring. Unless a signal, for example, too much product, turns them off.
What is RNA splicing?
In eukaryotes, the RNA transcript may be modified before it is sent to the cytoplasm to be translated into a protein. This process is known as RNA splicing or post-transcriptional processing. From an evolutionary standpoint, this ability to edit the message allows greater variety of expression from a finite set of genes. Current research indicates that most eukaryotic genes are interrupted or discontinuous, that is, the mRNA produced from the DNA sequence has had intervening nucleotides removed.
Are some forms of cancer communicable?
Yes and no. Cancer cells arise from within the body. However, certain types of cancer may actually be caused by viruses, which may be transferred from one organism to another. Currently, it is estimated that viruses may play a role in as much as 15% of human cancers worldwide.
What is the function of the skeletal system?
The skeletal system is a multifunctional system. The skeletal system provides support, allows an animal to move, and protects the internal organs and soft parts of an animal's body.
Why was Stewart successful?
Like previous investigators, he supplied the cultured cells with sugars, minerals, and vitamins. In addition, he also added a new ingredient, coconut milk. Coconut milk contains, among other things, a substance that induces cell division. Subsequent research identified this material as cytokinins, a group of plant hormones, growth regulators, that stimulate cell division. Once the cultured cells began dividing, they were transplanted on agar media, where they formed roots and shoots and developed into plants.
What is Müllerian mimicry?
Fritz Müller, 1821–1897, a German-born zoologist, described a phenomenon in 1878 in which a group of species with the same adaptations against predation was also of similar appearance. This phenomenon is now called Müllerian mimicry. Müllerian mimics include wasps and bees, all of which have similar yellow and black striped patterns that serve as a warning to potential predators.
Do different fungi rot different parts of trees?
Most fungi that rot wood in standing trees are basidiomycetes. The group that includes mushrooms, fleshy shelves, and rigid brackets. Most wood rotters attack only one or two related species of trees. Conifers and deciduous trees are more prone to fungi damage. Many wood rot fungi damage only specific parts of a tree. For example, Ganoderma lucidum and Heterobasidion annosum specifically rot roots and are rarely found in higher sections of trees. Sirena unicolor and Climacodon septentrionale are common in high sections of trees and are rarely found in the roots. Laetoporus sulphurus and Fomitopsis pinicola are found in all sections of a tree except the smallest branches of their host.
Why are mass extinctions a part of evolution?
A mass extinction is named for a time period in which at least 60% of the living species present become extinct over a period of 1 million years. Mass extinctions are considered biological catastrophes because of the relative speed and range of their effects. The loss of so many species allows surviving populations to exploit their adaptations in new ways. They can adapt to new parts of the environment without facing competition from other species. Certain organelles Example: chloroplasts and mitochondria within eukaryotic cells share a number of similarities with bacteria. Because of this, scientists surmise that early versions of eukaryotic cells had symbiotic relationships with certain bacteria. The eukaryote provided protection and resources, while the prokaryote specialized in converting energy, either sunlight or chemical, into forms that could be used by the eukaryotic cell. Sugar or ATP, thus the term endosymbiote, meaning shared internal life. Similarities between chloroplasts, mitochondria, and free-living bacteria include genetic material. All have DNA in chromosomes. Protein synthesis, all are capable of making proteins. Energy transduction, all are capable of acquiring and using energy to perform reactions.
How do fish swimming in a school change their direction simultaneously?
The movements of a school of fish, which confuse predators, happen because the fish detect pressure changes in the water. The detection system, called the lateral line, is found along each side of the fish's body. Along the line are clusters of tiny hairs inside cups filled with a jelly-like substance. If a fish becomes alarmed and turns sharply, it causes a pressure wave in the water around it. This wave pressure deforms the jelly in the lateral line of nearby fish. The deformation moves the hairs that trigger nerves, and a signal is sent to the brain telling the fish to turn.
What is bioprospecting?
Bioprospecting involves the search for possible new plant or microbial strains, particularly from the world's largest rainforests and coral reefs. These organisms are then used to develop new phytopharmaceuticals. There is some controversy as to who owns the resources of these countries. The countries in which the resources reside or the company that turns them into valuable products.
Which conifers in North America lose their leaves in winter?
Dawn redwood trees, Metasequoia, are deciduous. Their leaves are bright green in summer and turn coppery red in the fall before they drop. Previously known only as a fossil, the tree was found in China in 1941 and has been growing in the United States since the 1940s. The U.S. Department of Agriculture distributed seeds to experimental growers in the United States, and the Dawn redwood tree now grows all over the country. The only native conifers that shed all of their leaves in the fall are the bald cypress, Taxidium distichum, and the larch, Larix larsina.
What is the significance of Darwin's study of finches?
In his studies on the Galapagos Islands, Darwin observed patterns in animals and Plants that suggested to him that species changed over time to produce new species. Darwin collected several species of finches. The species were all similar, but each had developed beaks and bills specialized to catch food in a different way. Some species had heavy bills for cracking open tough seeds. Others had slender bills for catching insects. One species used twigs to probe for insects in tree cavities. All the species resembled one species of South American finch. In fact, all the plants and animals of the Galapagos Islands were similar to those of the nearby coast of South America. Darwin felt that the simplest explanation for this similarity was that a few species of plants and animals from South America must have migrated to the Galapagos Islands. These few plants and animals then changed as they adapted to the environment on their new home, eventually giving rise to many new species. Evolutionary theory proposes that species change over time in response to environmental challenges.
Which industries release the most toxic chemicals?
As of 2001, the metal mining industry released the most toxic chemicals. Although overall its output was less than that for the year 2000. Of the total 6.15 billion pounds, 2.8 billion kilograms of material released on and off-site by industry, food production. Commonly thought of as a mostly environmentally benign industry, was responsible for 2%. 125 million pounds or 57 million kilograms, or about 8 times as much as the coal mining industry.
What are nucleic acids?
DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are nucleic acids. Nucleic acids are molecules comprised of monomers known as nucleotides. These molecules may be relatively small, as in the case of certain kinds of RNA, or quite large. A single DNA strand may have millions of monomer units. Individual nucleotides and their derivatives are important in living organisms. ATP. The molecule that transfers energy in cells, is built from a nucleotide, as are a number of other molecules crucial to metabolism.
What is the oldest known fossil flower?
The fossil of the world's earliest known flower was discovered in 1986. This flowering plant is from the 120-million-year-old Kunwara fossil beds found near Melbourne, Australia. The fossil flower was an important find because all the parts of the flower were attached to an intact plant. The fossil resembles a small black pepper plant, less than 1 in 3 centimeters long. Paleobotanists believe that this plant represents an ancestral type of flower.
What is a better source of energy, fat or sugar?
Fats contain 9 kcal/g, while carbohydrates average about 4 kcal/g. Fats are useful for storing energy, but sugars are easily broken down during metabolism. Nutritionists and diet gurus are debating which source of energy is better. The absolute answer lies in individual situations. For example, babies require fat in their diets for both energy 24 and to build a healthy nervous system. Middle-aged adults quite often take in too much fat for their metabolic requirements. As one ages, average energy needs decrease by 5% per decade.
What is the optimum thickness for specimens to be examined under a microscope?
Biological and medical techniques require specimens of 1 to 50 micrometers, with the usual thickness being 4 to 5 micrometers for examination under a light microscope. Since an electron microscope has greater resolution, it requires thinner sections of 20 to 100 for biological specimens. Smear preparation is another standard technique used to prepare slides.
Whose experiments showed that non-lethal bacteria could be transformed into lethal bacteria?
In 1928, an Army medical officer, Frederick Griffith, 1878 to 1939, was trying to find a vaccine against Streptococcus pneumoniae. In the course of his work, he found that there were two strains of the bacteria, one that had a smooth coat S but was lethal. The other form had a rough coat R, which was non-lethal when injected into mice. He decided to investigate what would happen if he injected both heat-killed S bacteria and live R bacteria into mice. To his surprise, the mice injected with this combination died. Upon closer examination of the blood, living S bacteria were found. Something had occurred that transformed the non-lethal R bacteria into S bacteria. Subsequent experiments throughout the 1940s attempted to find the identity of the transforming factor, which was eventually found to be DNA.
How does smoking affect the DNA of lung cells?
Smoking appears to alter gene expression in lung cells. The proteins produced by the bronchial cells of smokers show increased synthesis of genes, which can eventually be carcinogenic. It appears that the expression of these important genes that control cancer development, cancer suppression, or airway inflammation varies according to the number of years spent smoking. The good news is that 2 years after a person stops smoking, his/her gene expression levels begin to resemble those of people who have never smoked.
What is a stem cell?
Stem cells are undifferentiated cells, meaning that they do not have a specific function that are capable, under certain conditions, of producing cells that can become a specific type of tissue. Stem cells present in adult humans are found in bone marrow and other tissues, such as fat. However, most research interest is focused on stem cells present in fetal tissue, which, in a laboratory setting, can reproductively divide indefinitely and be stimulated into becoming a variety of different cell types. The potential benefits of stem cells have made the research of these cells an exciting research topic.
How much water does an average sponge circulate during a day?
A sponge that is 4 in, 10 centimeters, tall and 0.4 in, 1 centimeter in diameter pumps about 23 qts, 22.5 L, of water through its body in one day. To obtain enough food to grow by 3 ounces, 100 g, a sponge must filter about 275 gal, 1000 kilograms, of seawater.
What is Gray's manual?
Gray's Manual of Botany, first published in 1848 by Asa Gray, 1810 to 1888, under the title Manual of the Botany of Northern United States, was the first accurate and modern guide to the plants of eastern North America. The publication contained keys and thorough descriptions of plants. The 8th, and centennial, edition was largely rewritten and expanded by Merritt Lyndon Fernald. 1873 to 1950, and published in 1950. This edition was corrected and updated by R.C. Rollins and reprinted in 1987 by Dioscorides Press.
What are the major features shown by all vertebrates?
Animals in the subphylum Vertebrata are distinguished from other chordates by several features. Most prominent is the endoskeleton of bone or cartilage. Centering around the vertebral column, spine or backbone. Composed of separate vertebrae, showing internal metamerism. A vertebral column combines flexibility with enough strength to support even a large body. Other vertebrate features include, 1. Complex dorsal kidneys, 2. A tail, lost via evolution in some groups. Extending between the anus, 3. A closed circulatory systems with a single, well-developed heart. 4. A brain at the anterior end of the spinal cord, with 10 or more pairs of cranial nerves. 5. A cranium, skull, protecting the brain. 6. Paired sex organs in both males and females. and 7, 2 pairs of movable appendages fins in the fishes, which evolved into legs in land vertebrates.
How do cells store energy?
Plants and animals use glucose as their main energy source, but the way this molecule is stored differs. Animals store their glucose subunits in the form of glycogen, a series of long, branched chains of glucose. Plants store their glucose as starch, formed by long, unbranched chains of glucose molecules. Both glycogen and starch are formed through the chemical reaction of dehydration synthesis, and both are broken down through the process of hydrolysis.
What is the largest group of vertebrates?
The largest group of vertebrates is fishes. They are a diverse group and include almost 21,000 species, more than all other kinds of vertebrates combined. Most members of this group are osteocytes, or bony fishes, which include bases, trout, and salmon.
Do humans really need cholesterol?
Cholesterol is an important component in the cell membranes of animals. Cholesterol, a lipid-based molecule, steroid, actually has two functions: 1, to help stabilize the membrane. 2, to maintain membrane flexibility as temperature changes. Normally the human body is capable of producing all the cholesterol it needs. Dietary ingestion of excess saturated fats and cholesterol is currently thought to be the source of the plaque that builds up in arteries and can cause heart attacks and strokes. Dietary cholesterol can be found in all animal sources, including shellfish. Plant phytosterols have the same function as cholesterol found in animals, but they do not affect the human body in the same way.
What are the functions of the cell membranes?
Cell membranes define and compartmentalize space, regulate the flow of materials, detect external signals, and mediate interactions between cells.
What is epigenetics?
Just as the term epidermis refers to the layer of the skin above, or beyond the dermis, the term epigenetics describes non-genetic causes of a phenotype. An example is genetic imprinting.
What are the main components of chloroplasts?
Chloroplasts have outer and inner membranes, which are in close association with each other. They also have a closed compartment of stacked membranes called 46 grana that lie inside the inner membrane. A chloroplast may contain 100 or more grana, and each grana may contain a few to several dozen disc-shaped structures called thylakoids that contain chlorophyll on their surface. The fluid that surrounds the thylakoid is called the stroma.
What types of variations are there in plant stems?
Plant stems show variation in size and shape. In addition, some plants have modified stems. For example, the strawberry plant has stolons or runners, which are horizontal stems that grow along the surface of the ground. Iris plants also have horizontal stems called rhizomes. Rhizomes are large, brownish, root-like structures found just below the surface of the ground. The rhizomes store food and can spread to form new plants. White potato plants also have rhizomes. The rhizomes of the white potato plant end in large, round structures called tubers. The tubers are the part of the plant that we call potatoes and eat. Tendrils and twining shoots of the morning glory and sweet potato coil around objects and help support the plant.
What is antisense technology?
Antisense technology involves targeting a protein at the RNA level as a means of regulating gene expression. The technology involves segments of DNA that are complementary to specific RNA targets. When the modified DNA binds to the RNA can no longer produce a protein. Antisense technology is a new technique in cancer therapy. Human trials are ongoing on small cell lung cancer and leukemia. Since cancer is a disease that can be characterized by excessive production of a particular protein, antisense technology works by interfering with the RNA that produces the protein.
How do cells respond to steroid hormones?
Progesterone, estrogen, testosterone, and glucocorticoids are steroid hormones that are lipid-based signaling molecules. After entering a target cell, the steroid hormone binds to a receptor protein and starts a cascade of events that ultimately activates, turns on, or inhibits, turns off, a specific set of genes.
Who is considered the founder of experimental zoology?
Abraham Tremblay, 1710 to 1784, a Swiss scientist, is considered the founder of experimental zoology. Much of his research involved studying the regeneration of hydras.
What are the stages of mitosis?
Mitosis involves the replication of DNA and its separation into two new daughter cells. While only 4 phases of mitosis are often listed, the entire process is actually comprised of 6 phases. Interphase involves extensive preparation for the division process. Prophase, the condensation of chromosomes, the nuclear membrane disappears. Formation of the spindle apparatus, chromosomes attach to spindle fibers. Metaphase, chromosomes, attached by spindle fibers, align along the midline of a cell. Anaphase, the centromere splits and chromatids move apart. Telophase, the nuclear membrane reforms around newly divided chromosomes. Cytokinesis, the division of cytoplasm, cell membranes. And organelles occur. In plants, a new cell wall forms.
What cheeses are associated with fungi?
The unique flavor of cheeses such as Roquefort, Camembert, and Brie is produced by members of the genus Penicillium. Roquefort is often referred to as the king of cheeses. It is one of the oldest and best-known cheeses in the world. This blue cheese has been enjoyed since Roman times and was a favorite of Charlemagne, king of the Franks and emperor of the Holy Roman Empire, 742 to 814. Roquefort is made from sheep's milk that has been exposed to the mold. Penicillium roqueforti and aged for 3 months or more in the limestone caverns of Mount Comelu, near the village of Roquefort in southwestern France. This is the only place true Roquefort can be aged. It has a creamy, rich texture and a flavor that is simultaneously pungent, piquant, and salty. It has a creamy white interior marked by blue veins. The cheese is held together with a snowy white rind. True Roquefort is authenticated by the presence of a red sheep on the emblem present on the cheese's wrapper. Penicillium camemberti gives Camembert and Brie cheeses their special qualities. Napoleon is said to have christened Camembert cheese with its name. Supposedly the name comes from the Norman village where a farmer's wife first served it to Napoleon. This cheese is formed of cow's milk and has a white, downy rind and a smooth, creamy interior. When perfectly ripe and served at room temperature, the cheese should ooze thickly. Although Brie is made in many places, Brie from the region of the same name, east of Paris, is considered one of the world's finest cheeses by connoisseurs. Similar to Camembert, it has a white, surface-ripened rind and smooth, buttery interior.
How can organisms with just one set of chromosomes reproduce?
An organism that is haploid, one set of chromosomes, can reproduce by mitosis to produce more haploid cells or a multicellular haploid organism. This is typical of some algae and fungi.
What is respiration?
Respiration is the exchange of gases, oxygen and carbon dioxide, between an animal and its environment. There are three phases to the process of respiration: gas exchange 1, breathing. When an animal inhales oxygen and exhales carbon dioxide. 2, transport of gases via the blood. Circulatory system, to the body's tissues, and 3, at the cellular level. When the cells take in oxygen from the blood and in return add carbon dioxide to the blood.
Who was Alfred Russel Wallace?
Alfred Russel Wallace, 1823 to 1913, was a naturalist whose work was presented with Charles Darwin's 1809 to 1882, at the Linnean Society of London in 1858. After extensive travels in the Amazon basin, Wallace independently came to the same conclusions as Darwin on the significance of natural selection in driving the diversification of species. Wallace also worked as a natural history specimen collector in Indonesia. Wallace, like Darwin, also read the work of Thomas Malthus, 1766 to 1834. During an attack of malaria in Indonesia, Wallace made the connection between the Malthusian concept of the struggle for existence and a mechanism for change within populations. From this, Wallace wrote the essay that was eventually presented with Darwin's work in 1858.
Why did sexual reproduction evolve?
The appearance and maintenance of energetically expensive traits like sexual reproduction will occur only if there is a net benefit to the fitness of individuals with those traits. There are costs to sexual reproduction, for example. Finding a mate can demand considerable time and energy. More importantly, individuals engaging in sexual reproduction only pass on half of their genes to each offspring. So their fitness is half that of asexually reproducing individuals. Scientists have examined the costs and benefits of sexual reproduction and determined that it is most likely to have evolved as a way to maintain genetic diversity. Experiments have shown that populations in erratic environments or those who are reproductively isolated from the rest of their species are at an advantage when they reproduce sexually rather than asexually. By being able to mix and match alleles, individuals within these populations can maintain genetic diversity and phenotypic variation, expanding their toolkit as a hedge against an unpredictable future.
How do animals know who is the boss in the herd?
Determining dominance can be overt or quite cryptic. It may involve a physical challenge and some kind of stylized fighting. Or it may rely on body language, like a change in the way a dog wags its tail. Sometimes animals determine dominance by cues too subtle for human observers to recognize.
How has plant classification changed over the years?
The earliest classifications of plants were based on whether the plant was considered medicinal or was shown to have other uses. De re rustica by Cato the Censor, 234 to 149 BCE. Lists 125 plants and was one of the earliest catalogues of Roman plants. Gaius Plinius Secundus, 23 to 79, known as Pliny the Elder, wrote Historia Naturalis, which was published in the 1st century. The book was one of the earliest catalogues of significant plants in the ancient world, describing more than 1,000 plants. Plant classification became more complicated as more and more plants were discovered. One of the earliest plant taxonomists was the Italian botanist Cesalpinus, 1519 to 1603. In 1583 he classified more than 1,500 plants according to various attributes, including leaf formation and the presence of seeds or fruit. John Ray, 1627 to 1705, was the first botanist to base plant classification on the presence of multiple similarities and features. His Historia Plantarum Generalis, published between 1686 and 1704, was a detailed classification of more than 18,000 plants. The book included a distinction between monocotyledon and dicotyledon flowering plants. The French botanist J.P. de Tournefort, 1656 to 1708, was the first to characterize genus as a taxonomic rank that falls between the ranks of family and species. De Tournefort's classification system included 9,000 species in 700 genera. The Swedish naturalist Carolus Linnaeus, 1707 to 1778, published Species Plantarum in 1753. It organized plants into 24 classes based on reproductive features. The Linnaean system of binomial nomenclature remains the most widely used system for classifying plants and animals. It is considered an artificial system since it often does not reflect natural relationships. During the late 18th century, several natural systems of classification were proposed. The French botanist Antoine Laurent de Jussieu, 1686 to 1758, published Genera Plantarum. The tome Prodroma Systematis Naturalis Regna Vegetabilis was started in 1824 by the Swiss botanist Augustin Pérème de Candolle, 1778 to 1841, and completed 50 years later. Another Genera Plantarum was published between 1862 and 1883 by the English botanists George Bentham, 1800 to 1884, and Sir Joseph Dalton Hooker, 1817 to 1911. Charles Darwin's, 1809 to 1882, ideas on evolution began to influence systems of classification during the late 19th century. The first major phylogenetic system of plant classification was proposed around the close of the 19th century. Die Naturlichen Pflanzenfamilien, the natural plant families. One of the most complete phylogenetic systems of classification and still in use through the 20th century. Was published between 1887 to 1915 by the German botanists Adolf Engler, 1844 to 1930, and Karl Prantl, 1849 to 1893. Their system recognizes about 100,000 species of plants, organized by their presumed evolutionary sequence. Systems of classifications were also developed during the 20th century. Some works focused on groups of plants, especially flowering plants, rather than all plants. Charles Bessey, 1845 to 1915, was the first American scientist to publish a system of classification in the early 20th century. Cladistics is one of the newest approaches to classification. It is often defined as a set of concepts and methods for determining cladograms, which portray branching patterns of evolution.
Why do birds flock?
The old adage about safety in numbers certainly holds true for flocking birds and other social groups. Flocks, herds, schools, and others provide a refuge for the young and an anti-predator advantage for all. The simultaneous movements of a large number of prey are apt to confuse predators and decrease the likelihood that they will capture any one individual.
How widespread is the use of GMO crops?
As of 2002, more than 120 million acres, 40 million hectares of fertile farmland were planted with GMO crops. The acreage is confined to 4 countries, United States, containing 68% of the total acreage. Argentina, 22%, Canada, 6%, and China, 3%.
What does pH mean when applied to soil?
Literally, pH stands for potential of hydrogen and is the term used by soil scientists to represent the hydrogen ion concentration in a soil sample. The relative alkalinity acidity is commonly expressed in terms of the symbol pH. The neutral point in the scale is 7. Soil testing below 7 is said to be acidic, soil testing above pH 7 is alkaline. The pH values are based on logarithms with a base of 10. Thus, a soil testing pH 5 is 10 times as acidic as soil testing pH 6. While a soil testing pH 4 is 100 times as acidic as soil testing pH 6.
What is fermentation?
Scientists have theorized that fermentation was the process through which energy was first harvested from organic compounds. Fermentation evolved before Earth's atmosphere contained free oxygen. Aerobic respiration differs from fermentation in that the products of glycolysis enter the Krebs cycle rather than being used to form lactic acid or alcohol. Fermentation must have predated the appearance of oxygen in the atmosphere, which occurred over 2.5 billion years ago. Fermentation is thus an ancient process and occurs normally in microorganisms that live in the absence of oxygen.
How has biotechnology helped thalidomide become popular again?
The drug thalidomide was developed during the 1950s. It was found to be effective in combating the symptoms of morning sickness in pregnant women. Unfortunately, if taken during the first trimester of pregnancy, thalidomide causes serious limb deformities because it interferes with the normal growth process of the fetus. Recently, it was discovered that the drug also has the ability to interfere with the replication of HIV in human cells. Thalidomide also may prevent the weight loss commonly associated with AIDS. Thalidomide actually has two mirror image forms, chirality, only one of which causes birth defects. Researchers hope that by using biotechnology they will be able to separate the forms and return thalidomide to their arsenal of disease-fighting chemicals once again.
What is the function of the endomembrane system?
The endomembrane system allows macromolecules to diffuse or be transferred from one of the components of the system to another.
What are the major groups of echinoderms?
There are 6 principal groups of echinoderms, from the Greek terms echina, meaning spiny, and derma, meaning skin. 1, class Crinoidea, sea lilies and feather stars. 2, class Asteroidea, sea stars. 3, class Ophiuroidea, basket stars and brittle stars. 4, class Echinoidea, sea urchins and sand dollars. 5, class Holothuroidea, sea cucumbers. And 6, class Concentricycloidea, sea daisies, which live on waterlogged wood in the deep sea and were first discovered in 1986.
Why is electrophoresis important for biological research?
Gel electrophoresis has become an important tool to identify DNA molecules, genetic differences between different species of plants, and animals have been identified using gel electrophoresis. Protein molecules that differ by a single amino acid can be identified. This process is useful for determining the genetic fingerprint of an individual.
What is unusual about the way the emperor penguin's eggs are incubated?
Each female emperor penguin, Aptenidites fursteri, lays one large egg. Initially, both sexes share in incubating the egg by carrying it on his or her feet covered with a fold of skin. After a few days of passing the egg back and forth, the female leaves to feed in the open water of the Arctic Ocean. Balancing their eggs on their feet, the male penguins shuffle about the rookery, periodically huddling together for warmth during blizzards and frigid weather. If an egg is inadvertently orphaned, a male with no egg will quickly adopt it. 2 months after the female's departure, the chick hatches. The male feeds it with a milky substance he regurgitates until the female returns, now padded with blubber. The females take over feeding the chicks with fish they have stored in their crops. The females do not return to their mate, and own offspring, however, but wander from male to male until one allows her to take his chick. It is then the male's turn to feed in open water and restore the fat layer they lost while incubating.
What is the biological basis for DNA fingerprinting?
DNA fingerprinting, also known as DNA typing or DNA profiling, is based on the unique genetic differences that exist between individuals. Most DNA sequences are identical, but out of 100 base pairs of DNA, 2 people will generally differ by 1 base pair. Since there are 3 billion base pairs in human DNA, one individual's DNA will differ from another's by 3 million base pairs. To examine an individual's DNA fingerprint, the DNA sample is cut with a restriction endonuclease, and the fragments are separated by gel electrophoresis. The fragments are then transferred to a nylon membrane, where they are incubated in a solution containing a radioactive DNA probe that is complementary to specific polymorphic sequences.
What is a base pair?
The phrase base pair is a way of describing the length of DNA. Since most DNA is double-stranded, every nitrogen base containing nucleotide on one strand of the molecule is paired with a complementary base on the other strand. Thus, 10 base pairs refers to a segment of DNA that is 10 nucleotides in length and 2 strands in width.
How do steroid hormones differ from non-steroid hormones?
steroid hormones such as estrogen and testosterone enter target cells and directly interact with the DNA in the nucleus. Non-steroid hormones such as adrenaline generally do not enter the target cell but instead bind to a receptor protein found on external cell membranes. This then causes a sequence of metabolic effects.
What are the non-chylomates?
The non-chylomates are animals that lack a body cavity, a chylome, and include the sponges, jellyfishes, and simple worms.
What is cloning?
A clone is a group of cells derived from the original cell by fission, one cell dividing into two cells. Or by mitosis, cell nucleus division with each chromosome splitting into two. Cloning perpetuates an existing organism's genetic makeup. Gardeners have been making clones of plants for centuries by taking cuttings of plants to make genetically identical copies. For plants that refuse to grow from cuttings, or for the animal world. Modern scientific techniques have greatly extended the range of cloning. The technique for plants starts with taking a cutting of a plant that best satisfies the criteria for reproductive success, beauty, or some other standard. Since all of the plant's cells contain the genetic information from which the entire plant can be reconstructed, the cutting can be taken from any part of the plant, placed in a culture medium having nutritious chemicals and a growth hormone. The cells in the cutting divide, doubling in size every 6 weeks until the mass of cells produces small white globular points called embryoids. These embryoids develop roots, or shoots, and begin to look like tiny plants. Transplanted into compost, these plants grow into exact copies of the parent plant. The whole process takes 18 months. This process, called tissue culture, has been used to make clones of oil palm, asparagus, pineapples, strawberries, Brussels sprouts, cauliflower, bananas, carnations, ferns, and others. Besides making highly productive copies of the best plant available. This method controls viral diseases that are passed through normal seed generations.
How do salmon find the way to their spawning grounds?
Scientists do not know exactly how a salmon remembers the way back to its native stream. After an ocean journey possibly lasting several years and covering several thousand miles. They agree, however, that salmon, like homing pigeons, appear to have an innate compass or search recognition mechanism that operates independently of astronomical or physical signs. Some scientists theorize that this internal compass uses the infinitely small electrical Voltages generated by the ocean currents as they travel through the Earth's magnetic field. Others believe that the salmon's homing mechanism may take its cues from the varying salinities of the water or specific smells encountered along the journey.
How are ribosomes different from other organelles?
Ribosomes differ from most other organelles in not being bound by a membrane.
What plants are erroneously called mosses?
Not all plants called mosses are bryophytes. Irish moss, Chondrus crispus, and related species are actually red algae. Iceland moss, Cetraria islandica, and reindeer moss, Cladonia rangiferina, are lichens. Club mosses, genus Lycopodium, are seedless, vascular plants, and Spanish moss. Tillandsia usneoides, is a flowering plant in the pineapple family.
What are truffles and where do they come from?
Truffles, a delight of gourmets, are arguably the most prized edible fungi. Found mainly in Western Europe. They grow near the roots of trees, particularly oak, but also chestnut, hazel, and beech, in open woodlands. Unlike typical mushrooms, truffles develop 3 to 12 in 7.6 to 30.5 centimeters, underground, making them difficult to find. Truffle hunters use dogs and pigs that have been specially trained to find the flavorful morsels. Both animals have a keen sense of smell and are attracted to the strong, nut-like aroma of truffles. In fact, trained pigs are able to pick up the scent of a truffle from 20 feet 6.1 meters away. After catching a whiff of a truffle's scent, the animals rush to the origin of the aroma and quickly root out the precious prize. Once the truffle is found, the truffle hunter, referred to in French as a trufficulteur, carefully scrapes back the earth to reveal the fungus. Truffles should not be touched by human skin, as doing so can cause the fungus to rot.
How many types of muscle tissue are found in vertebrates, and what is the function of muscle tissue?
Vertebrates have 3 types of muscle tissue: 1. Smooth, 2. Skeletal, and 3. Cardiac muscle. Muscle tissue, consisting of bundles of long cells called muscle fibers, provides the capability of movement for the organism or for the movement of substances within the body of the organism.